This is a survival curve chart for human rabies. During the months of incubation, the line remains flat, but at the moment of symptom onset, survival drops vertically to near absolute zero. Once neurological symptoms appear, medical intervention fails. Antivirals and life support cannot stop the disease. Rabies achieves this lethality through a coordinated, low-profile neuroinvasion. It ignores rabid tissue destruction, focusing entirely on reaching the central nervous system undetected. The virus sabotages the immune system, hijacks internal transport networks, and metabolically starves neurons from the inside out. To understand why this infection is incurable and how we can intercept it before the point of no return, we must track the pathogen's geographic path from the initial bite to the brain. An infection begins when a bite or scratch deposits the virus deep within skeletal muscle tissue. At this entry point, the virus replicates at extremely low levels. Because the virus does not enter the bloodstream, it bypasses systemic immune sensors. Without viremia, a diagnosis is impossible during this early stage. This period of localized replication is the incubation phase, which typically lasts between 1 and 3 months. The molecular breach occurs at the neuromuscular junction. Viral particles bind to nicotinic acetylcholine receptors, which act as the specialized gateways into the host's motor neurons. The virus has now crossed into the nervous system, successfully evading the body's systemic immunological alarms. Once inside the motor neuron, the viral G protein facilitates entry into the cell interior. The virus does not drift randomly. Instead, it hijacks the cell's internal microtubule transit system to move toward the spine. This hijack is precise. The viral P protein docks directly into the host's motor protein, cytoplasmic dynein. Through retrograde axoplasmic transport, the dynein physically hauls the virus up the length of the axon and toward the central nervous system. This process moves at a velocity of 5 to 100 millimeters per day. It is a slow but relentless race toward the brain. By utilizing this transit system, the virus turns the body's neural architecture into an unmonitored highway. When the virus reaches the central nervous system, viral replication increases exponentially. To prevent the inflammatory response that would open the blood-brain barrier, the virus avoids destroying brain tissue immediately. The virus instead creates a localized safe zone through active immune subversion. Rabies forces the infected neuron to produce specific death ligand proteins, FAS-L and HLA-G, and move them to its surface. When the host's T cells attempt to attack the neuron, they bind to these new receptors and immediately undergo apoptosis, or programmed cell death. This mechanism induces an artificial state of immune privilege, allowing the virus to replicate without local interference from white blood cells. Because the blood-brain barrier remains intact, systemic antibodies cannot cross from the blood into the brain to assist. The virus achieves lethality by weaponizing the brain's own protective barriers to create an impenetrable fortress. Wild-type rabies does not kill through widespread cell death. It does not burst the neurons. The severe symptoms are the result of virus-induced metabolic dysfunction. The virus triggers a massive overproduction of inducible nitric oxide synthase. The resulting clouds of nitric oxide fill the interior of the neuron. This excessive nitric oxide starves the cell's mitochondria, cutting off the ATP energy production required for the neuron to function. This oxidative stress leads to the physical swelling and degeneration of the axon, which halts the transport of essential proteins. The host's neurons are not technically dead, but they are paralyzed and starved. With their metabolic systems failing, they can no longer transmit signals or maintain basic cellular functions. This microscopic dysfunction manifests as the severe behavioral changes of symptomatic rabies. In furious rabies, which accounts for 80% of cases, the viral load in the limbic system causes extreme agitation, aggression, and hydrophobia. In paralytic rabies, the infection of motor pathways leads to a quiet, ascending paralysis. The virus then utilizes anterograde transport to move centrifugally away from the brain and into the salivary glands. These behavioral symptoms ensure the virus is shed into the saliva and transmitted through a bite just as the host perishes. Once symptoms appear, the virus is irreversibly entrenched behind the blood-brain barrier. Survival depends on exploiting the slow incubation period before the virus enters the central nervous system. Post-exposure prophylaxis begins with wound washing and the administration of human rabies immune globulin, which works to neutralize the virus locally in the muscle tissue. An accompanying vaccine series then forces the body to build systemic antibodies to hunt down any remaining viral particles. To increase global access, the WHO recommends moving from intramuscular to intradermal vaccine administration. This chart compares the costs. Intradermal administration uses a smaller dose to achieve the same result, offering a 60 to 80% cost reduction. Globally, the most effective way to eliminate human rabies is to stop the pathogen at its source through mass dog vaccination programs. Rabies employs a highly specialized evolutionary strategy. Since the virus is inaccessible to standard medicine once established in the brain, our only true defense is molecular interception and global prevention.